I'm Kelly Shepard. I'm a scientific program manager in the BPN Biologics program. And I'm going to be talking to you today along with my colleague, Lauren Friedman, all about our program and how you can become a part of it. Lauren, next slide. So just so everyone's aware, this webinar will be recorded and made public. It'll be on the website. We're also going to send PDF copies to everybody who registered as attendees. There is a Q&A box in Zoom for you to post all your questions. We are going to address questions at the end. You can enter the questions at any time though, and then one of our presenters with us will read the questions so we can go through them with everybody. If you do have project-specific questions related to specifically what you might want to do within this program, we encourage you to contact our email address instead of posting them in the Q&A, since again, this is all a public discussion. Next slide. And today we're gonna walk you through just our general program overview. Lauren's gonna talk to you about the award mechanisms that are available and funding opportunities, as well as preparing your application. And I will end by talking about our contract resources that are available to people in this program, followed of course by our Q&A. All right, so our program supports biologics, and by that we really do mean the wide spectrum of modalities that are available within biologics and including novel and emerging therapies. We currently support projects in pretty much everything seen here, and we do see interest in new and emerging technologies, and all of them are things that we want to support within our program. Next slide. And the goal for our program is to accelerate biologics-based therapeutic development for nervous system disorders by advancing therapies through the early phase clinical trials. And we do this by providing both grant funding and in-kind resources to bridge critical gaps currently available to academic and small business entities. As part of this, we're looking to help you de-risk a project and actually create outside investment to enable the later stage phases and bring these novel drugs to market. And we're hoping to identify sort of the best ideas and methods within the NIH research community through this funding opportunity, and of course, supporting it with the associated infrastructure. Through all of these goals for our program, we preserve the institution's intellectual property in order to facilitate licensing. And this is to say that NIH does not take any stake in the IP that's brought into the program. This is what we refer to as a trans-IC, or Institute and Center program. There are 9 different institutes who participate and who will fund projects. And so when we say nervous system disorders, it could fall under any one of these institutes or centers. And we strongly encourage you to go to the website to review the specific interests for the institutes and centers involved to determine where your project might fall. So the project support is generally divided into several buckets. There is grant funding for work that is done by PI-directed research activities, and it includes funding for any contractors or consultants that you would bring in that, and fund through your grant and manage. There are also contract research services that we provide at NIH. So we provide you access to industry standard non-clinical manufacturing and early phase clinical services. And these are available for you to take advantage of in the program, and you would work with NIH staff to use those resources. We also have a large collection of consultants that provide assistance from in all the drug development efforts. They have extensive experience in a wide number of topic areas related to biologic therapy development, and particularly industry experience in order to help guide the project. And at the end of the day, all of this culminates in a project team, a multidisciplinary project team that supports the PI, that supports the entire effort and is tailored to meet the project needs. Overall, we provide up to 5 years of support and access to all of the resources focused mostly on lead optimization, IND enabling activities, and then the first in human clinical trial. And again, no matter who is doing the work, whether it's one of our contractors or something that's being directed within your lab, the PI's institution retains the right to existing IP and is assigned rights to any new IP developed in the program throughout this time. Slide. And I'm going to hand it over to Lauren to introduce herself and talk about the different award mechanisms.
Thank you, Kelly. So I'm Lauren Friedman. I'm another scientific program manager in the BPN Biologics program. And like Kelly mentioned, I'm going to walk us through the type of mechanism that the program uses, our funding opportunities, and then some tips on preparing your application. So starting with the award mechanisms, we have two companion funding opportunities, and they both use a phased award mechanism. And for those of you that aren't familiar with phased awards at NIH, these basically have two different separate awards under one grant with an interim checkpoint. And this is for appropriate for things like drug development, where NIH would want to make sure the project is sufficiently de-risked before moving on to the larger and more expensive studies in the second phase. So our two funding opportunities are UG3, UH3, which we'll get into later, and this kind of covers all applicants. And then the U44 is an SBIR fast track. So each has two phases, roughly similar amounts of time. And they're also milestone-driven awards. So this means that you'll propose aims, but we'll turn them into milestones, the type that are used in drug development. So as you can see here, milestones will be specific and rigorous and quantitative. And so as we prepare milestones together with the PI, we'll look to develop go, no-go criteria. Did you hit this criteria with your experiment? If yes, proceed to the next milestone. So all projects, regardless of where they're starting, and we'll talk about that in a bit, will start in the first phase, UG3 or U44 phase 1. All applications will include proposed work for both of the phases. I talked about the milestones and then this interim checkpoint. So we'll be working through milestones as you go through the first phase, but there is kind of this distinct point where there will be an evaluation on whether the project's sufficiently de-risked and ready to advance to the next stage. And I should just note this terminology, phase 1, this is not referring to clinical trials, the common phase of clinical trials, this is just the SBIR term. So the other aspect that might be unique to this for some of the folks on the webinar is we use a cooperative agreement as opposed to a grant. For those who haven't had or applied to cooperative agreements before, it's important to point out some of the differences between a traditional grant. And so the biggest difference is the involvement of NIH staff in a really collaborative nature with the PI team. And it involves very regular and sometimes frequent communication, because as we'll talk about soon, there are a lot of moving parts to these projects. So this table is for your reference, just the difference between, or key differences between cooperative agreements and grant awards. So in our program, the cooperative agreement comes not just with funding, but also contract resources and consultants that Kelly will talk about later. As we talked about, NIH staff involvement is substantial versus minimal post-award with your more typical grants. Communication is also much more frequent than is typically involved with the grant through emails and meetings, and then reporting. So we'll talk about this in a bit. We'll convene a team that'll meet regularly, most likely bi-weekly, and so there'll be informal project updates, but also more formal presentations, annual milestone progress presentations, and then your typical annual NIH progress reports. As we go through these slides, we have here reference to our NOFOs, that's Notice of Funding Opportunities, and the section with the relevant information. So let's talk a little bit about the highly collaborative project teams we form around each awarded project. So we use this term lead development team, and that consists of the PI and their research team. This could be people in the lab, collaborators at your institution or outside of your institution, grant-funded consultants. So if you want to bring on your own consultants, or let's say you've been working with a drug development consultant, you can use grant funding to cover those individuals and they can join the team. Same goes for CROs, these contract research organizations. If there are certain vendors, service providers you'd like to work with, you can cover it through your grant as a sub-award. And all these individuals will join the LDT at different times. The bolded ones are constant. These will be the ones that join the meetings all the time. On the other side, you have NIH program staff, NIH contracted consultants. So every project will get assigned at least one drug development consultant that we'll talk about later. And then eventually, if you choose to bring on or propose work through NIH CROs, eventually a project manager and study directors from that CRO will join the lead development team. So this is a multidisciplinary team. It's assembled for every awarded project specific to the needs of that project. Like I mentioned, we typically meet bi-weekly. These are virtual meetings of 1 to 2 hours. And the important thing here is the regular communication because we're dealing eventually with a number of different study sites. Maybe we have ongoing work at your grant-funded CRO and several of our NIH CROs, and it's all happening simultaneously. Maybe there's work going on to prepare for a regulatory package. And so there can be times where there's a lot happening, and these meetings are important to just stay on top of things, understand where things are working, where they're not, troubleshoot, and make sure things are moving forward in the milestones. And PI expectations, again, because this is quite different than your typical grant, it's important to mention that this LDT meeting will be led by the PI, and we expect that there'll be regular updates and also prompt communication or responsiveness with NIH. And all this really is in support of the PI-driven work. At the end of the day, this is the PI's project. They are driving the project, and this team we build around it is there to support it and hopefully advance things forward. So we'll next talk about our 2 funding opportunities, which have been recently reissued, and the links to both are here. So first thing is, what's the difference between these 2? They're fairly similar, but there are a couple of key differences. So UG3, UH3, as mentioned, this is open to all eligible applicants, whereas U44 is an SBIR, which is only eligible or only open to eligible US small businesses. And as a fast track, that means it's a combined or seamless phase 1, phase 2. The duration for both is up to 5 years. There are some slight differences in the UH3 phase, but basically it's 2 years for the first award phase and then 3 to 4 years for the second award phase. The budget for the UG3, UH3 isn't limited, but should be reasonable and reflect project needs. If it does exceed $500,000 in direct costs in any year, there are additional requirements, which we can touch on briefly. As for the SBIR, there's no hard cap, but these are some of the guidances that rarely exceeds these amounts in total costs. And there's no specific requirements for the large budget approval. But if in either case you expect that you'll go way over, you'll exceed some of these guidelines, we really encourage you to just contact us early. And you'll hear us say this often throughout the presentation. Contact program staff, the earlier you can, the better, so we can start working on this pre-approval process. Okay, so these next few slides will talk a bit about the scope of what is covered through the 2 NOFOs. So projects can enter at one of 2 stages, either starting with a promising lead or lead agents or a single candidate. So we use this term discovery stage to mean you have a promising lead and you still need to do lead optimization. Or you can start the program at the development stage. You've identified a single candidate. You're nearly ready to go into IND enabling, but you probably still have some preparatory work to do, be it developing assays and And we'll go into that in the next slide. So this lists the criteria for discovery stage projects. There should be some preliminary in vivo data. Assays and models should be scientifically sound and available and really ready to go and validated if you plan to use them as part of your lead optimization. If you find that you don't have these yet, there are other funding opportunities throughout the different participating institutes and centers that can cover this. And no more than 2 years should be required for lead optimization. Development stage, it should be a clinical candidate with all the suitable characteristics listed here. And again, if you look at section 1D in our NOFOs, it'll go into more detail on the entry criteria. An additional criteria is that patents should already be filed, or if you're not there yet, at least an intellectual property strategy should be in place. And this is important. You might have a great idea, but perhaps you haven't thought about the IP, how you would protect this, how it's differentiated from other people's technology. And one resource is to contact your technology transfer office at your universities. Companies, I think, are more familiar with this, but we do want to see that some kind of plan is in place, even if patents haven't been filed or applied for. Okay, so next is the in-scope R&D activities. So this is taking the same discovery stage with the lead or development stage with the candidate, and starting in either lead optimization or you've completed it and you need to do some prep work to go ahead with IND-enabling studies. For all projects, the minimum aim or the minimum goal is to reach IND submission within 5 years, the end of 5 years. If your project is more advanced or if feasible, the funding opportunities also support a first-in-human clinical trial. And this is optional, but if the timelines are feasible, we highly encourage that you include this as well. But importantly, reviewers are really going to take a look at your project feasibility. So you want to make sure what you're proposing makes sense in terms of timelines for reaching these 2 points. So as mentioned before, all projects must start either in the UG3 phase or the U44 phase 1. And here's some sample activities for discovery stage we've talked about, lead optimization. You can also do some of this preparatory work, but it should not exceed 2 years. If you're coming in at the development stage, your project's more advanced, so we'd expect that your first phase wouldn't exceed more than 1 year. And some of the work you can do are it could be pharmacodynamic biomarker development and qualification. It could be analytical development, initial process development, bioanalytical development, all in preparation for the next phase of work and pre-IND meeting. And then here's this award phase transition. So this is kind of like the major checkpoint. If projects advance, then these are the activities, really all the IND enabling type of work, IND submission, and then the optional clinical trial. And again, you can check out section 1E and the NOFOs for more. A word about clinical trial activities. So through the programs, we support first-in-human clinical trials. And so this just gives you an idea of what is in scope. So it could be healthy subjects or patient populations, single dose or single ascending dose, placebo-controlled or open label studies. And then, these are the typical endpoints for the early stage programs. And we very strongly encourage you, if you're thinking of doing a clinical trial, you have any idea that you want to do a clinical trial, to reach out to us. We can talk through, potential feasibility of the proposed time frame. And then importantly, if you have interest in using one of our contractors to conduct your first-in-human study, it would be good to talk about your patient population and if the contractors support that population currently. Okay, and then next were non-responsive activities. So just to make really clear what's in scope, what's outside of scope. And so again, what's in scope is specifically biotherapeutic development for these stages, lead optimization through first-in-human clinical trials. And then there's a list of things that are here and in the NOFO that are non-responsive. Importantly, NINDS and all of our participating institutes and centers have different funding opportunities to support all of this work. Of note, I'll just mention for small molecule therapeutics, we have a sister program, BPN small molecules, and these are the two NOFOs. But you'll find on our website some of these funding opportunities to support this work. Okay, so moving on to preparing your application. So as I mentioned, our NOFOs, that's notices of funding opportunities, are now posted, and these are the links here. Next receipt date is January 27th, 2025. So there's plenty of time to prepare and get in touch with your questions. And then as you would typically typically the application guide, read the NOFOs very carefully, and contact us early. This email goes directly to the BPN Biologics program staff. And when in doubt, you can reach out to us first, and we can always connect you with the appropriate institute center contact from one of our participating ICs. So we just wanted to pull out a couple aspects that are unique to this application. This is not a comprehensive list. So again, look at the links for the complete instructions, but couple things to be prepared to work on. One is an intellectual property strategy, as we talked about. That's one of the entry criteria, and so that's a one-page required attachment. New for people who've applied to these funding opportunities before, this is a new required attachment. It's a Gantt chart. I'm going to show an example of that in the next slide. Target product profile. So this is included in the research strategy. Therapeutic agent profile. This is a table you'll create that summarizes the known characteristics of your lead or candidate. Also, it goes in the research strategy. Another new piece is the testing funnel. This is only for discovery stage projects, and this is basically a way to summarize the proposed work from lead optimization to candidate selection. And finally, the data sharing and management plan. So there are example tables and charts available on our website, and you can also find out more information in the NOFOs. Okay, so the two new elements, the Gantt chart, which many of you are probably familiar with, we show this example on our website, and this is just meant to show all of the proposed activities through the duration of your proposed funding period. And it should be by year and by quarter. And importantly, is using some kind of color coding pattern, some way to differentiate who's going to run what study. That's really important for program staff and the reviewers to know who's going to do what, because the types of activities are actually described differently in the application. So the Gantt chart is a really good way to make it clear who you propose to do what. And then, as I mentioned, the testing funnel is a schematic or a flow chart to show your starting point, and then all the different in vitro assays and animal studies you'd run to get down to one candidate. And then you'd also show your go, no go criteria, your proposed no go criteria. And again, this is only for discovery stage projects. Okay, a couple other tips to point out in the research strategy and budget. So importantly, your application should only focus on a single nervous system disease. Even if it would be applicable to other diseases, the application itself must only focus on one disease, and it should be within the mission of one of the participating institutes and centers. As I mentioned, you'll summarize your eventual marketed product, what that'll look like in a target product profile or the TPP table. There's an example of that on the website. You'll need to specify your project's entry stage. You have to just call out, I'm starting at the discovery stage, I'm starting at development stage. And then as we talked about discovery stage projects, you should expect to reach candidate selection within 2 years, and you should expect to complete the UG3 or phase 1 within 2 years. Development stage, because they're more advanced, would be 1 year. Summarize your known properties in the therapeutic agent profile, which examples are on the website. present supporting data and study descriptions in compliance with NIH guidance on rigor and reproducibility. So NIH has developed a lot of helpful tools and guidances on this. So encourage all of you to look at that. And then include a budget for every year of the proposed project, including both award phases. Okay, so how to address CROs and consultants. So basically the NIH contracted CROs and consultants versus the grant funded CROs and consultants. So there's a couple key differences. So one, how they're funded. So grant funded would be through a subaward. You bring on whoever you like, propose to work with them, have a subaward. With the BPN contractors, this is covered through NIH contracts, and so it's not part of the budget. No cost. It comes in addition to the grant funding. Selection for grant funded, of course, is by the applicant, but for the BPN Biologics contractors, it's with NIH staff after award. And Kelly's going to talk about that process a bit. So how do you describe these in the application? So the grant funded should be identified in the application. So the names and the proper descriptions of the company or bio sketches for the consultants, if necessary. That's not needed for BPN contractors. You might go on our website and see CROs and consultants, but you don't need to pick any out. And in fact, you shouldn't pick any out and put in the application. So you might just mention, I propose to do this activity through an NIH CRO. Expenses in the application include for grant funded, do not include in your budget if you want to do NIH CROs, and then detailed study designs in the application. So just like any other experimental work, if it's a grant funded CRO, describe it with proper justification. If it's a BPN contractor, you can describe it at a high level and maybe talk about the deliverables, but it's not expected that you have the study design down because that'll be developed in collaboration with your lead development team. So high level, it does not need to be detailed in your application. Okay, so with that, we'll hear more about contractors from Kelly.
Thank you, Lauren. All right. We have a few different contract resources to tell you about. If you want to go to the next slide. So the first group are consultants. And so we have a large number of consultants available to help our projects. You can see some of the companies that we've pulled from. We also have a wide array of independent consultants, and they cover all the major areas of biologic therapy development that would be needed. We're constantly pulling in people with expertise in different modalities and different parts of it. So we have quite a number of consultants available, and they all have extensive industry and regulatory experience, either in specific modalities or broadly. And their job really is to join the project teams. We as NIH staff actually assign them post-award, and this is again after looking at your application, what who you're bringing to the table, and who might be needed to really best support the project. And they make technical recommendations to the team. They provide guidance based on their experience and knowledge for both milestones and the overall development strategy. And they assist our research and development contractors in study designs and package preparation and everything kind of that's needed by the project teams. We do cycle our consultants on and off the teams, during the funding period, really to make sure that you're getting the best stage specific guidance for your project. Next slide. And then we provide a number of R&D contractors or CROs we set up this access as they're all industry standard manufacturing, non-clinical and early phase clinical services. So they can cover anything that you want to propose in your project as long as it falls within the scope that Lauren talked about, the lead optimization and the IND enabling and then the clinical trial. But their use is optional. So you can look at what you feel very confident in doing through your own grant funding in your own lab or through your own CROs, and then you can propose to do other activities through our CROs. It is important to know that the actual selection of the specific contractor who's going to do whatever work is needed occurs after the grant is awarded and in consultation with the project team that consists of your entire team and consultants brought on, the NIH consultants and NIH staff. So it is a very collaborative process by which we determine what work is needed and exactly what work is needed for the project, and then actually selecting the contractors. And then no matter what, all of the work done by our contractors, they assign the rights to any new IP developed from that work to you. So we ask that you come in with what we refer to as unencumbered IP, or your project can actually reach commercialization, and we're going to make sure that you leave this program with unencumbered IP so that you can take it on to later phases. Next slide. And so these are, again, just example services provided by our CROs, everything within the non-clinical space, your bioanalytical and PD assay development and qualification, PK and tox studies, biodistribution work, both non-GLP and GLP environments, and then manufacturing of pretty much every modality that we see. And this is the full campaign from early analytical and process development clear through GMP runs and clinical supplies if applicable for your project. And then our actual contractors are listed at the top, our prime contractors. It's important to understand that the way we run our contract in order to ensure we can actually support all of the various modalities and all of the projects that we see is we have three main prime contractors, we call them. They really serve as integrators. They hold the main contract, and they are encouraged to do what they can in-house and then to subcontract for activities that they can't do in-house. And this gives us a central group with whom we're working, and a central group coordinating all these activities while simultaneously getting us access to niche resources necessary. And you can see some example subcontractors currently working on projects supporting BPN projects. Next slide. We also have a separate contract that covers the first-in-human clinical studies. And this group will actually engage in study planning and even writing a protocol for you. They can be brought on before the IND submission in order to assist with that. And then ultimately the coordination, conduct, and data management and reporting for the trial itself. The current contractors available are Duke and PPD. And again, they are also allowed to subcontract, though they are much more likely to run these studies in-house. And as Lauren mentioned, we really encourage you to contact us before making plans for a first-in-human through our contractors, particularly if your first-in-human trial is won't be able to be in healthy subjects. We really want to make sure we walk through and understand what the needs will be so that we can give you the best possible advice for your project. Next slide. All right. So we've thrown a lot of information at you about how these NOFOs work and how the program works and everything. So I'm going to offer some tips for next steps. These are tips. These are not required activities. However, this is usually sort of an order of operations that will hopefully get you the best guidance as you develop an application. The first really is to read through the NOFOs. I know that they're long and there's a lot of information, but the NOFOs and the application guide really do provide a lot of the details and help you organize what's the kind of information I need to provide. For those of you who are familiar with the NOFOs that were part of this program up to this point, they have been rewritten some. There are a couple new elements that Lauren highlighted, like the Gantt chart and the testing funnel. The majority of the NOFO is the same general idea. However, we did attempt to clarify many of the things that have been questions over time. So please read through them as they provide very clear descriptions of what it is that's needed for different types of projects. After you've had a chance to look through the NOFO, if you feel like this is definitely the program that you're ready for, I would work through at least a draft budget and your IP considerations. Talk to your tech transfer office if you have one. They are often excellent at helping you with the IP strategy and just overall laying out where your product sits within the world and the different types of considerations you need. And then on the budget side, it really is key that you determine if your budget will exceed that 500K indirect cost in the first 2 years for the UG3, UH3. Generally speaking, though, it's also a good idea to work through your budget, because if you're very close to that 500K and you make a change that bumps you over, all of the rules apply for getting the additional approvals and paperwork that's needed. So this is something to work through early and to kind of understand where you sit and what you're likely to do with the potential budget. And then you are welcome to contact us. Again, we recommend at least 8 weeks in advance of the intended receipt date so that any advice, recommendations, or discussion can actually be incorporated. But please contact us to set up a pre-submission call, and we will talk to you about your project, help maybe clarify anything about the program that needs clarification. If you can, when you do that, I highly recommend you send a draft specific aim statement. If you don't, we will probably ask you for one as our first question. It usually is a very good way to orient us to the project and give us kind of the high level things we need to make sure that we can advise you on fit and overall use of the resources. It's important to note we cannot tell you if your application is good or is going to be well reviewed, but we can give you an idea of the fit and the appropriateness for the program. All right, so thank you so much for joining us today. And remember, please, I'm going to give everybody a minute to put any questions they might have in the Q&A so that we can go over them in just a moment. If you do have project-specific questions where you'd like to discuss more detailed elements of your project, please email us. As I just said, pre-submission calls are an excellent idea and can help clarify a number of things. Additionally, our website is being updated, and a lot of information about the NOFOs and about the program can be found on the BPN Biologics website. We will actually send you a notification when the fully updated website is ready to go, and I highly suggest you check it out. Particularly a lot of the examples that Lauren mentioned for a lot of the documents that are necessary will be found on that website. And we will be updating it over time as more resources become available. All right, I think we have most of our questions in the chat, though there is one in the Q&A. I'm going to turn it over to Elena, one of my colleagues, to help walk us through our questions.
Yeah, I'll be just reading them as they were coming in the easiest way. So the first question in the Q&A is whether this U44 mechanism is considered for USBAR only or STTR as well.
So this mechanism is SBIR.
These mechanisms. And whether these BPN grants available with biotech startups with academic PIs as a co-founders, or are they only available to academic PIs?
No, absolutely. Many of the biotech companies that apply and work with were started by academic PIs. So it would be up to the applicant whether they wanted to apply through the company through the U44 mechanism or the UG3 mechanism. And if anyone has questions, of course, reach out.
The BPN Biologics program support the repurposing of an agent if the repurposed agent can be patented and protected by intellectual property rights?
Typically, it would be something novel. I think it would depend. I think we would want to understand, is there a novel formulation? What is the IP position? So let's take that on a case-by-case basis.
Will first-in-human trial is supported only if it starts before the end of the 5th year?
Yes. So if you propose a first-in-human clinical trial, then you should anticipate completing it within the 5-year funding period.
Completing or starting? The question was like, does it have to be starting before the end of the...
You should anticipate, you should build out the timeline so that you could complete it within the 5-year period, not just start it.
If we apply for discovery stage, do we need efficacy data on the non-optimized lead before applying?
Yes, you need some preliminary data. In vivo, it should be target engagement and efficacy. As to what is considered efficacy, then that's something we could discuss. But the lead should be well characterized, even if you anticipate doing further optimization.
Do you allow collaboration between two labs from two different institutes for these opportunities? If multiple PIs are involved, would you prefer senior or middle career investigator to be the lead PI for the project team? This is mostly relating to the frequent meeting requirement and interactions with NIH.
So we do allow the multiple PI awards, and we would leave it up to the project teams. That would be a discussion on with the team on what makes sense and who would be the one to be at all the meetings at the primary point of contact. The universities also should be talking about IP arrangements and the where the IP would ultimately come from. So again, it would be a discussion between the applicants or the multiple PIs.
I think this question was answered, but I still will read it. Do you allow collaboration between the university PI and the small business?
Yes. It depends on how you structure it. So the first decision would be, would the primary applicant or PI be from the company versus the university? And that might inform which mechanism you apply to. But in either mechanism, you could sub-award to the other entity.
Okay. Contract-related question. Do you have CROs that support non-human primate studies?
Yes, all of our CROs can support non-human primate studies.
Thank you. And can a project have a major overseas component, example Belgium, in which the IP is held by a foreign company and some developmental work would be done there or at a foreign academic institute?
It depends, one, on if it's a business is the applicant or an academic institution is the applicant. I think that kind of detail we'd want to discuss on a case-by-case basis. So please reach out.
Yeah, though it is important to note that the SBIR must be a US-based company. So this would only work for the all institution or UG3H3 mechanism. There are some additional considerations that are a little specific. So yeah, if you reach out, we can talk through your exact situation.
Thank you.
How do you define freedom to operate?
In general, so we're not asking for a full freedom to operate analysis, which can be really quite extensive and cost a lot of legal fees. That's something that will be done much further down the road. What we're interested in knowing is, do you foresee, are there any known blocks to being able to develop the technology? Are you using a component of the technology that is owned by someone else? Maybe it's a capsid. You kind of want to have an understanding of who owns all the components and that no one's going to say, hey, wait a minute, you can't go ahead and manufacture this through the funded award. So just something to start thinking about early.
Thank you. What level of details we need for NIH BPN service, and how do we find what they can do?
So,
oh, sorry, go ahead, Kelly.
oh no, you're good. But to use the NIH contractors, you don't really need to provide significant detail. You can say that we're planning to do, for instance, our GLP tox studies through the BPN CRO. You'd be identifying the approximate timing that you expect those to occur, right? What's going to lead up to that, which sort of milestone or specific aim it relates to, and that'd be about all you need to say about it. You are welcome to look at the website and see some more information about them. I will tell you they were vetted to come into our program because they can, either in-house or through a subcontractor, perform absolutely every study needed to support this program. We both have not come across a case where they cannot support a project, but also part of our due diligence is making sure once we actually see applications that the kinds of things that we are getting are the kinds of things that they can support. So we're in constant communication with them. There's nowhere where you can find really specific or detailed lists about what they do. I would just assume that they can do anything that you're asking of them in the application as long as it falls within scope for this program.
Thank you. Our research needs and CRO needs might continue to evolve. Is it possible to add new grant-funded CROs after award?
Doing so is a process. I can't say no. It is possible to go through a process to add funds and things. It is difficult, lengthy, and not guaranteed. We highly recommend that you have mapped out your plan and the CROs you choose and the work that they're going to do is well defined within the application. And quite frankly, that's one of the reasons for having contracted resources is it's going to be easier for us to support evolving research needs via those contracts. So it's not that we can't do it, it's that there's a number of consideration and steps involved. One of the roles of the project team and the set of experts is to really make sure that any evolving project needs are met and the best way to do it. And that includes analysis of whether we should do it via grant or contract funding.
Okay, next one. Given the timeline of review, like general deadline and award date in December, is it accurate that all funded work should start after award notice?
Yes, that's correct. So part of the process from submission and then your typical review, and then there's a period where NIH staff will work with you on developing the milestones. That'll then go into the notice of award. As soon as the notice of award is issued, work can start.
This initiative, what are examples of cell products as a therapeutic agent?
Sure. So a recent project that's been awarded is an iPSC-derived neuron therapy to be injected into the brain for epilepsy. But we would consider any type of cell product that fits within the mission of one of the participating institutes. So it would depend on the disease, but we are always seeing new types of therapies. I think that's the exciting thing with biologics. Even if it wasn't listed, maybe we don't know about that type of therapy yet. We might see that. So any specific questions about cell therapy projects, please reach out.
I think this is more like comment than a question. It would be extremely helpful if the BPN Biologics program can provide a few examples of funded proposals.
They are listed on the website, and I know we didn't include them in this particular presentation, but they are on the website. And so you can see their title, disorder modality, the current lead and institution.
How many applications are funded per cycle?
So we currently, through this NOFO, have 2 cycles per year. And it is a very competitive program because of the resources that are offered. So it could be a few projects a year, but it really varies.
How many, this is a relating question. What success rate of U44 BPN Biologics applications can one expect? And the same for UG3, UH3.
Yeah, I'll kind of just reiterate, it is a highly competitive program. So it's just several a year.
And it is good to note that the UH3 and U44 are similar in their competitiveness and in number of applications received, right, and opportunities. So there is not a significant difference between the two in terms of success rate.
This was the last question in the chat, so guess we are done.
Great. Thank you so much to everyone that attended. Like we said a million times, email us with your questions. We're happy to talk. We're happy to hear about your projects, answer your questions. And